how can information transfer between the genetic code and then to the translation of proteins? The central dogma was that the information would also pass from the DNA to some form of RNA, and then from RNA to the translation of some form of protein. So this is the central dogma of how information would be passing down. And also, this idea was mentioned in that Watson and Crick paper that was published in '53. And they were saying, they were just speculating about how this transfer of information would happen. But they said, it's just a speculation. Maybe it wasn't that. But we know that this happens, DNA from RNA to protein. Now, another thing that can happen too in case of retroviruses is that you can have your genetic material encoded in RNA as well. It doesn't have to be DNA. Right? You can have viruses that are perfectly fine with RNA. They code for all the proteins, then infect the host, and then some of them will transfer that information into a DNA by reverse transcription. That means if you have the RNA and it becomes a cDNA, you're not transcribing it. Transcription means DNA to RNA. A reverse transcription means from RNA to DNA. Okay, so that can happen, and then from DNA you can go back to RNA and then down to protein. Or you can have the translation straight from the RNA that has the code. It can go straight from RNA back to protein like this, and it will never be transformed into DNA. Okay, an example of a virus that uses the retrovirus, the reverse transcription, is HIV. It's an RNA virus. It will infect the cell and insert the DNA into the nucleus. And that DNA is going to find a piece of the host DNA, is going to find a piece that is going to insert itself in there. Okay, so this is a case in which the central dogma is not really the norm. So we have the RNA going to DNA, and then from there, later on, when it starts to transcribe its gene, when it's not silent, it will go back into RNA, go into the cytoplasm, and then express its proteins for replication. Then if you want an example of a virus that does not do that and is only on RNA form, that is the vesicular stomatitis virus. Okay, this is a cytoplasmic virus. This is the nuclear. Cytoplasmic virus is RNA virus, negative strand RNA. It gets in there, starts to make protein right out of the RNA. Right out of its genetic material, starts to make protein. Okay, now, how can you know about these structures? We know the DNA is, usually that double helix. Now, they can form other forms of structures. Mainly, we see these structures in RNAs, because RNA is a single-stranded, it's not double-stranded. If you have this structure that is single-stranded, and it happens that it has bases that will match with the other side of this strand, they can loop around this string and form these hairpin-looking structures. Okay. Now look, this one really looks like a little hairpin with a loop in there, because in this case here, the bases are not matching. So here you have restrained, because CA is not going to match with C here. So then you have this little bubble that is just not attaching to each other. But if you have a continuous match, you just get this hairpin that is a little loop like that. You can have with this bubble loop here or just without the loop. But they do form these secondary structures. Even if it is single-stranded, they can form these secondary structures. Mainly happening with RNA. Okay, so here is say RNA can have many, many hairpins, and those hairpins will make a particular characteristic of a certain RNA. Okay, so it will form its own structure based on how it will fold in itself. So it's important to know the similarities and the differences between RNA and DNA. We know that which one is single-stranded. RNA is single-stranded, DNA is double-stranded. RNA has a uracil, DNA has a thymine. And then the sugar in the RNA is the ribose, with both feet in place, right? DNA is the deoxyribose. Okay. Now this we went over in the lab, right? How to measure DNA, how to work with DNA. Now, so I'm just going to brush a little bit faster here, but it is also part of the lecture of the course. It's not only the lab. It's a practical way of how to work with the DNA. So does everybody remember what wavelength DNA will absorb? 260, right? And the protein would be at 280. Then if you want a pure sample of DNA, you have to have a higher level of 260 over 280 ratio, right? We went over into a lot of detail in the lab for that. And so if you have any questions about how each one of these practical approaches will work, just ask. Okay. Now, the other one was this centrifugation, in which one of them is this gradient centrifugation, in which you have media that is this gooey substance, right, that you're going to put the DNA through, and you're going to separate it based on the density, not on the weight, but on the density. And what I said, like fat floats, and then if you have hard muscle, it sink, right? And anyway, you get your equilibrium of buoyancy, and that's how you do the separation with the gradient centrifugation. So once you have all the molecules separated by density, by the density equimolar part, the density equilibration part, you just poke a hole in the bottom and get your different samples and check which one has your DNA. Now, the other form of centrifugation is the sedimentation velocity centrifugation. That means the bigger the mass, the faster it will get to the bottom. Okay, so this just has to do with how quick that thing can get to the bottom of the centrifugation. We also went into a lot of details of the denaturation and renaturation of the DNA, that if you heat it up, you separate those hydrogen bonds, and then if you cool it down, you're going to get those hydrogen bonds annealing back together, right? And you know that you can stipulate, you can more or less calculate how much is the ratio that you have on this DNA molecule. You calculate the ratio of A to T's and C to G's, because if you have a lot of hydrogen bonds in the case of C to G bonding, you are going to need higher temperature to separate these two molecules of DNA. You need to put a lot more energy into breaking 3 hydrogen bonds than into breaking 2 hydrogen bonds. Was that idea clear in the lab? Everything's cool? Yeah? Okay, the melting temperature, it has to do with half of the molecules melting at that temperature. Okay, we mentioned everything like that. And also we went over the comparison, the sequence comparison. Does everybody understand how that is done? Different species, let's say the chimpanzee and the human. Want to go over this again? Yes? So here we have 2 species. You have chimpanzee DNA, and you have human DNA. This is the double helix. Okay? So now you want to see how is it that they compare. Is this DNA exactly the same as that one? I mean, how can we compare one species to another? This is a very crude method of doing that. So what you do is that first you denature, increase the temperature. You denature this molecule, so now you have single-stranded. Okay, it's not double-stranded anymore. And now you put them together in this test tube. You just mix them together, and slowly you lower the temperature. Now remember that base pairing is very specific. And remember the way that you're planning for primers and for cloning, you usually get a small piece of unique sequence of the DNA, and that's how you plan your primer. That's how you do PCR, right? So annealing is very specific. So whatever is in this sequence that is matching to the other sequence is going to anneal. Okay, so let's say here you have a perfect match. Okay, so here we have like A, T, C, G, and then on the top here you have T, A, T, A, G, C. These are going to match perfectly. Now, if you start having differences between these 2 pairs, they're not going to match. Now, since this one will draw bigger than the other one, it's just going to be a loop here. Until they'll find later on down the line, you see something that will match again. Okay, so C, G, T, 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 that will match again. So if you look at that DNA and you see what is matching and what is not matching, you can calculate the proportion of how similar these two species are. Okay, all this here, let's say, is matching for 98% of the bases. So these species are very close to each other, right? So if it was matching something like 50%, you'll say that's very far apart. That's more like a human and a mouse. Okay, so this is how you do a base analysis of how similar the sequences are, these different species are. Okay, also we talked about how quickly the DNA will anneal in case of repetition. If you have bases that are repeated many, many times, like in this case here, 80, 80, 88 forever, the chances of them annealing very fast is very high. They will just zip up very quickly. Now, if you have another sequence that is more complex and is not a very uniform two-base repeat or one-base repeat, then you're going to have a little more trouble, or the DNA is going to take a little longer to match up those sequences, to match up those bases. So also when you compare how long the DNA parts will take to anneal to each other, you can also figure out on how complex the sequences are. Okay, we talked a lot about the in situ hybridization, how to probe specific parts of DNA. Right? I talked about how to, remember I talked about how to probe messenger RNA. Right? Messenger RNA has all this tail that is poly A tail, which you're going to see very, very soon. Then if I make a probe that is oligo-DT with lots of T's, it's going to anneal to all the poly A tails of the messenger RNAs. And then if you have some fluorescent probe on it, like a fluorescent molecule, maybe GFP, then we'll be able to see it. So you can locate specific areas by putting probes on the DNA. And you can also detect diseases or mutations or just the location of a particular gene. How to detect DNA by using radioisotopes, the Hershey-Chase experiment, like where the isotope went and how to trace it based on either heavy isotopes or radioactive isotopes. And we went over that on the experiment and also in the, as I had mentioned, the tracing of, in this case, RNA tag on a specific RNA. And then if we infect these RNAs in the virus, then if we infect the mouse, where is the virus going to show up? Okay. We can trace exactly where it's going. And this, we went through a lot of practicality on how to use this, right? The gel electrophoresis, and how the heavy, big DNA will stay on the top, has trouble going through these little seaweed fibers, and the small DNAs are just going to migrate quickly towards the positive electrode. We covered this a lot. So that's the end of chapter 10, so now we go to chapter 11.